So just to start, a little bit of history. As you can see, the the microbial collection story, I would say, in Italy really has very, very strong roots and foundations in Piedmont. We had, as a scientist in Piedmont, really the fortune and the privilege to have established already during the 50s a first kind of nucleus thinking about preservation and safeguarding of microbial diversity. This was really done at the level of the Biology Institute that at that moment started to store everything that had to be connected to the environment and to the mainly filamentous fungi with the environment. And that's why the work that was done before, at the end of the 90s, was really formalized with the institution of the Mycoteca Universitatis Tauriensis, that is the MUT that is one of the first collections in Italy that could really meet the parameters and the characteristics that is really into the specification of the culture collection. Okay, so in 1999 was established at the Department of Plant Biology at the University of Turin, and then in 2006 it got certified following the ISO 9001. You can see that immediately after it entered into the networks of the culture collection organizations like the World Federation and also the European Culture Collection. And then what has been happening is that through the, let's say, inspiration of the persons that were behind the MUT, and here I really need to extend my great thanks to my colleague Cristina Varese, that was the responsible of the Mycoteca Universitatis Tauriensis, that she was always very present and very enthusiastic about trying to push for the concept of the microbial diversity preservation and the microbial diversity safeguarding. And so what has been happening around 2010 is that she started to connect very closely to some of her colleagues, including me, that were spread around different departments working on microbial diversity, to try to establish a kind of network of collections that, let me say that at the moment, more than collections, were just tubes that were stored either in refrigerators or in -80 without any kind of organization or without any kind of, let's say, structure, but really to try to establish this concept of the microbial biopreservation in our region. What has been happening, as it was said before by Sarah, 4 departments actually put together their resources, and we established as a kind of entity within the University of Turin, the Turin University Culture Collection. The Turin University Culture Collection at this point was a little bit the organization that started to prime all the interactions with other entities and other organizations in Italy that were dealing with microbial biopreservation. And that's why in 2017, we established a joint research unit, MIRRI-IT, that was, let's say, formalized by the signature of an agreement by 5 founding members. Sarah mentioned them. I'm going to be showing it later. From this nucleus of 5 founding members, now we can proudly say that we have reached a national dimension, and we have 27 institutions that are actually part of the MIRRI-IT. And as a last let's say, event that has happened that is actually underlining the success of the establishment of the MIRRI-IT as a kind of network, a national network, was that after a lot of efforts and after a lot of energy that Christina put in and all the other members put in, we were able in 2025 to have the endorsement of Italy to enter into the MIRRI-ERIC. And so now MIRRI-IT is actually an official member of the network MIRRI-ERIC. Here is a little bit what I was telling you before. So you can see the spread all over Italy. We have the founding members that are the ones that are in yellow, orange, that are all belonging to the 5 entities that you see here with their logos. So the University of Turin, the University of Modena and Reggio Emilia, University of Perugia, the National Research Council, and the Ospedale Policlinico San Martino. So those were the ones that established the MIRRI-IT, and around them, we were able to grow and to reach the dimension that we had today. And so we can really say that MIRRI-IT has a powerful engine of scientific and technological innovation. What we do, as all the other collections around the world, is to really protecting, preserving, and safeguarding microbial biodiversity. We are collections of viruses, archaea, bacteria, yeast, filamentous fungi, and microalgae. And we are very much into the connection with the stakeholders. We have a very big collection of microorganisms of environmental, food, and veterinary interests. And so we are connecting very well with the industries that are working in these domains to offer solutions, or actually to create solutions to address some of the challenges that we are facing at the moment, but also to give opportunities for a business creation. And I would say that probably the most important event that has happened to us, again, through the opportunity that was given by the Ministry of Education to the infrastructures in Italy, was the presentation of a proposal that was called SUS-MIRRI within the Next Generation Fund. That was in 2022, and we have just finished, I mean, a couple of weeks ago, we had the final event here in Turin with a total budget of 17 million that was distributed around the consortium. With those money, what we have done was really give a big burst on the facilities. And so the laboratories of the partners participating in the network really had access to funds to acquire cutting-edge technologies and cutting-edge machines, edge machines for analysis, characterization, sequencers, but also for metabolic activities and all these kind of things, apart from the biopreservation and the storage, let's say, or the preservation of the strains. We have implemented services, and so now we have a list of services that is a common one for all the different partners participating in the project. And with the project, we were able actually also to somehow fill the gap between the state of the art that we had before in the Italian Microbial Culture Collection, reaching the MIRRI-ERIC standards. And so this is something that really helped us to structure and to reach a point where we can really say that we are up to speed in order to present ourselves as a reliable partner. This is just a screenshot of the website of the project. So you are more than welcome to visit it. And of course, there is also the version in English. I'm not expecting you to be able to read and to understand Italian, but there is also the version in English. So again, very welcome to have a look at it. Just a very brief introduction to what we have done in the SUS-MIRRI, because as you can see, SUSMIRI was a project that could not be really identified as a research project. It was a infrastructural project where we really defined work packages that could tackle some of the important aspects that needed to be addressed in order to strengthen and make stronger the infrastructure by itself. And so apart from the management, then we had a very, very important work package that was the work package number 2 regarding the strengthening of the culture collections and all the standards and the procedures behind the preservation of the microbes. Very important IT component with the work package number 3, where the people worked a lot in order to have databases, interoperability between the different databases, and dialogue between platforms in a way that this could be integrated in one Italian catalog of microorganisms. I want also to underline the very important work that has been done at the level of the development of bioinformatics workflows. So now we have in-house created pipelines for the analysis of metabarcoding, for instance, but also for the analysis of metagenomic data. And then it comes the work package where I was VP leader. Probably this is the only work package that could be identified as a research work package. That was the one focusing on microbiomes. We are going to be talking about it later, so I'm not spending any more time describing what we have done because we will have the possibility to go deep in the next slides. And then very important work packages regarding services and education. So here is a lot of courses that we have done. I'm going to be showing you some numbers just to give you an idea of what has been achieved during the implementation of the project, and then communication and dissemination. And again, some numbers will come in the next slides. Here is what SUSMIRI meant for us. A lot of effort from a number of researchers, technical, administrative staff, PhD students, more than 43,000 preserved characterized microbial strains that are now safely stored in our premises, an online catalog, more than 15 procedures for compliance regulations, 150 specialized technical scientific services, 40 plus training courses, 15 transnational access, program funded, 100 publications, and as you can see, 80,000 website visitors and 120,000 views in social media channels. So this was in a nutshell, basically what we were able to reach in the 3 years from 2022 to 2025. And here it comes now, the second part of my presentation. That is the one that is a little bit more technical, because I'm sure that you are very well aware of what is the actual situation regarding the understanding of the complexity of the ecosystems that we are dealing with when we are talking about environment, we are talking about agriculture, we are talking about food. So the challenge that we wanted to face, or that we wanted to address better in the SUSMIRI project, was really understand how, or first of all, if it was feasible or not to actually preserve whole microbiomes, and if we could use those microbiomes for further applications. And so I like to summarize the objective of this VP in the words that you see here in the screen, exploring the potential of microbiome as an ecosystemic solution, because I really believe that microbiomes, they have the potential to address a lot of different challenges that we are now facing in different ecosystems or in different contexts that are really close to our day life. I mean, think about the climate change, think about the desertification, think about the food waste. I mean, there are a lot of different, I would say, spaces that could be addressed with the potential that is inherent of the microbiomes. And the three, I would say, the three highlights that I would like to describe, that I would like to address also in the next slides, is really about developing standard operation procedures for microbiome sampling and analysis, because yes, there is a lot that has been done, but there is a lot of let's say dissipation. There is a lot of difference around the world in terms of protocols, in terms of procedures that are used for the sampling and also for the analysis. Then really the core of the work package was the protocols for the safe storage of microbiomes. So you will see that a lot of people put a lot of efforts in order to understand how this could be done. But then we were not happy just with the challenge of safely storing microbiomes, but we would also wanted to see if we could propagate them and reuse. So again, I would say that if we go from the first to the third point here in the slide, I mean, the level of complexity and the level of the level of the challenge is actually increasing, I would say probably also exponentially. Increasing exponentially, why? Because the approach that we wanted to give, or they wanted to take actually in the implementation of this VP, was really the One Health approach. You know, the One Health approach is an holistic approach where the health and the planetary health is really connected to every single, I would say, organism or individual that is present in this planet. Originally, it started with animals and humans, and the fact that, of course, we had issues related to the transfer of pathogens from animals to humans, and then there it was really initiated the idea of working at the level of the animals in order to protect the humans. But then the following development was also to include in this kind of holistic approach also the environment. And so now we have this picture that you see here that takes into consideration those elements, and only if those elements are in equilibrium, we can reach a kind of One Health and a planetary health, as it is called now. In order to follow this approach, you can see that the different units participating in the project, and here we had something like 15 to 20 units that were actually very, very engaged to work on this challenge, selecting ecosystems that were spanning from the environment. And so you can see the ones here in pale green, on organisms, I mean, considering also insects, let's say organisms, and of course the plants, and so humans and animals. And then we had the part that I'm going to be focusing also in the rest of the presentation, that is represented by foods. Fermented foods specifically, both solid and also liquid. So you can imagine that every single ecosystem that we had considered in the project was presenting specificities, challenges that us as researchers, we had to face in order to reach the final objective of the work package and of the project. Of course, we had different levels of arrivals, because in some cases we were stopping at the storage of the microbiomes and of the quality control of the microbiomes during the storage. In other cases, and most of them were related to foods, we also wanted to see if stored microbiomes could be reused. And here it comes, as I said before, the challenge of the propagation of the microbiome in order to be able to be used as an inoculum for the fermentation of the specific fermented food. This is a little bit, I just want to put here 3 slides before getting into the details and giving you an example of a little bit the path that we have followed and we have developed during the implementation of the project, just to give you like a kind of, again, first insight of the outcomes of this work package. The first one is about the standard operating procedures. Not only we have developed the standard operating procedures for sampling and analyzing all the ecosystems that you have seen in the previous slide. So here you have a snapshot of what we have deposited in the Zenodo database that represent both protocols for the sampling, but also protocols for the extraction of the DNA. But we didn't stop here because what we wanted to do was also to go into the validation of the SOPs. So this is to me a very, very important example of the collaboration and interaction that we had within the units that were working on the project, because we were able to validate the SOPs for foods, for soil and for water, establishing a kind of model in which some groups were responsible for collecting the samples, sending anonymized samples around in the other units, and then the other units, they were responsible for the extraction of DNA, and then this DNA was sent back to one unit where all the sequencing took place. So this was a very big effort that we were able to successfully address. And as you can see, I mean, at the beginning of this year, we had this paper that was accepted in Environmental Microbiome. It is open access, and so it is accessible by everybody. The second goal that we achieved was about the storage protocols. As I said, a lot of different ecosystems, a lot of different contexts that we needed to address. For each of them, we were able to design a specific protocol in order to successfully storage the microbiome. Here you see the annex, you see the number of the annexes that we had, because each of them represents one protocol for the specific ecosystem. Just a snapshot on some. I mean, here you have the first one on top is about the fermented legumes. So for tempest storage, so you can see here what it has been done. Three different ways of storing the microbiome: by dehydration, freezing, lyophilization, and then of course all the analytical methods that were related to the quality assurance of the storage protocol. On the bottom, you have the workflow for the preservation of natural way culture. So those are two examples of what we have done. Consider that all the 16, they actually present very practical information on how you need to work in order to be able to store your microbiome, following specific indications and following specific protocols. And then to finish was the story of the propagation protocols. As I said before, the propagation was not something that we have achieved for all the different ecosystems that we have investigated or that we have studied, because for some of them we did not see the need for a reuse or for a, let's say, a direct application or let's say real life application. So for 8 ecosystems, and as you can see from the slide, most of them were really related to foods like fermented sausages, table oils, sourdoughs, fermented legumes, and fermented beverages. We actually attempted the possibility to propagate the microbiome and to use the propagated microbiome as a starter culture for the production of fermented foods. And here you have 2 examples. You have the first example on top is about sourdoughs. where you can see that there is, I'm just going to be putting the, let's see if I will be able, yes, here. You can see this part is related to bacteria, this part is related to yeasts, and this is sourdough that is actually propagated at different times of storage. In one case, you have the possibility to actually maintain well the diversity. So actually in the fungal community, I mean, you see that there is a T12, there is actually a nice representation or a nice respect, let's say respect of the microbial composition. If we go to the bacteria, the situation was a little bit more complicated, because when we used the microbiome in the real life applications, you can see that in one case we could somehow have the populations present in the stored microbiome represented in the sourdough. In the other case, we were not successful. And in the bottom figure, here is a picture that is representing the volatilome of fermented sausages that were produced with different propagated microbiomes. And what I want to point out here is that look at the difference between the control, that was a spontaneous fermented sausage, with those that were obtained with the inoculation of the microbiomes, and with the one that was obtained by the backsloping. So the backsloping, that basically represents a piece of sausage that was stored at -80 for 1 year. So the inoculation of the backsloping really can be, let's say, reproduced with the preservation of the microbiomes in specific ways. All these abbreviations, I'm going to be telling you in a second exactly what they mean. But this to me was a nice result that we could obtain, because it could really clearly show that there is an opportunity here by storing microbiomes in a specific way and reusing them for successful fermentations. And here now it comes the applied, I would say, the applied research part, that is the work that we have been performing in my lab here. I'm already working for more than 20 years in fermented sausages, and so fermented sausages is a traditional product from Italy, but I would say from the Mediterranean area, that really needs attention and really needs a kind of special place in our culture, in our diet. What we have done as a preliminary work for the MIRRI project was really to screen a large diversity of fermented sausages in Italy. Here you see the number of sausages at the end of the maturation that we have collected: 24 samples from the north, 8 samples from the center, and 17 samples from the south. And you can see that only few regions in Italy were left out, because we could not actually get access to the samples. As expected, there is a huge difference in the formulation, in the technology, and in the maturing protocols. I mean, just looking at the presence of the garlic and wine as an ingredients, for instance, you can see how there is no standardization, in a sense that we cannot say that products from the north are using wine or products in the south are not using wine. No, the situation is quite complex, as we would expect, because again, fermented sausages is quite a traditional product. And so we have protocols, and we have also producers that they are using their own procedures, for which they are very, very jealous, and they don't want to communicate to the others. But the other thing is also about the certification. So as it happens in other countries in Europe, we have different levels of certifications. Here we just wanted to divide those that had certifications and those that did not have certifications, just to an additional point to, let's say, try to group or try to classify the products that we had analyzed. We did microbiological analysis. Honestly speaking, what we have seen was what we would expect, in a sense that we had very important, let's say, contributions that were given by groups of microorganisms that we know they are already very, very important in the maturation and the fermentation of fermented sausages. So you can see here the Staphylococcus and Cocuria, lactic acid bacteria, yeasts, total bacterial count. And then you have also the values of the water activity and the pH. So I would say that was nothing very striking coming out of this analysis. But of course, when we moved a little bit more deep and we went into the characterization of the microbiota of those sausages, here we started to see a lot of differentiation. So if the numbers are somehow stable, if we go down to the species level, we start to see really differences that they are more related to the specific products than the geographical positioning of the sample. Okay, but again, you can see that of course there is one member here that is very, very highly present. It is the Lactobacillus sakei, that we know it is an important lactic acid bacteria for the fermentation of fermented sausages. But in the case of the Toscana, for instance, in the sausages from Toscana, you can see that there is also another one together also with this sausage here, that is the Lactobacillus curvatus. That is the second one that we know it is very important in terms of participation in fermented sausage fermentations. More interesting is when, after the metagenomic sequencing, we started to reconstruct metagenome assembled genomes. So this is a picture describing the MAG distribution of the different species that we were able to reconstruct. So from the metagenomic libraries, we reconstructing genomes belonging to the different species. And to me, it was quite striking that in the sausages that we had collected throughout Italy, we had almost 25 different genotypes of Lactobacillus sakei and 20 of Lactobacillus curvatus. So this, and again, there is not really a distribution between the north, the center, and the south. So again, the situation is quite, let's say, dispersed in terms of presence or absence of specific marks. And you can see also how the different marks and the different species are actually present in the different fermented sausages. So this is actually underlining that fermented sausages, and I forgot to say that all the fermented sausages that we have analyzed are fermented sausages produced by spontaneous fermentation. So there are no starter cultures that are added in this spontaneous fermented sausages. Are a huge resource in terms of microbial biodiversity. And so imagine what all those microorganisms, both Lactobacillus sakei but also curvatus, can give as a kind of opportunity for the industry if they are properly characterized and if they are properly biopreserved. But this was, again, just the very beginning, because out of all this work, we selected 2 sausages that represented the systems that we wanted to preserve. Okay, so what we have done was to take those 2 sausages that in this next slides are going to be called as fermented sausage A and fermented sausage B. We applied different sample preparation methods before the storage at -80 degrees. Here you see what we have done. So we added first 30% of glycerol as a bioprotectant, and then we stored the fermented sausage as such. we stored a pellet obtained from the homogenization and the centrifugation of the solution obtained from the homogenization. So this was the pellet in a solid form. And then we also stored at -80 the first 10 times fold serial dilution. So as a kind of counterpart of the pellet, because in one case we had the solid part, and in the other case we had the liquid part. We stored it for 4, 7, 10, 12 months. After each time point, we towed the sample. We did microbiological analysis for vital counts, for viability counts, and then we did also metabolomic analysis, and we did also metagenomic analysis to understand how the microbiome was doing. The next step was to reuse the stored microbiome, and here, as I said before, the challenge was really the propagation. So what we have done was trying to use different strategies for propagation of the samples that we had stored at -80. And the propagation that we had performed was in 3 different liquid broths. One that was quite poor and minimal in nutrients. That was the nutrient broth. The second one was the brain heart infusion, very rich. And the third one was a meat extract that was basically resembling a little bit the characteristics, the physicochemical characteristics of the meat. The only difference was that in one case was solid, in the other case was liquid. And after the inoculation of the microbiome that was stored at -80, we incubated at 30 degrees, and we did the analysis at 6 hours and at 24 hours. In the next slides, I'm going to be presenting only the results of the 24 hours, and you will see that already there is a kind of very high complexity in the presentation of the data, because we have obtained a lot of stuff. Microbiological analysis, in order to understand how the counts were doing, also because, remember, we were storing for up to 12 months, and so we needed to understand what was staying alive and what was dying, but also molecular and chemical analysis and metabolomic analysis. Here is the snapshot regarding the preservation. So here you see counts. So those are the 2 sausages that I was telling you, sausage A and sausage B, and those are the trends in terms of yeasts, total bacterial count, total viable counts, coagulase negative Kochi and lactic acid bacteria. So you can see that the situation, especially for the sausage A, is quite stable, because the numbers are really staying quite constant, moving from the beginning to the 12 months. While in the sausage B, we have this weird trend, especially in the total viable count, that we don't know actually how to explain that during the preservation we had the growth of total viable count. So probably this is like a kind of mistake in the interpretation of the data that we have on the colonies that we have on the total viable counts and on the PCA. But for the rest, let's say, again, we have stability. You can see that in both cases we have a quite big difference between the populations of the lactic acid bacteria and the coagulase negative Kochi, and also the yeasts. So the yeasts, I can tell you, that are the ones that probably are suffering the most during the preservation. And also, if we take into consideration the different samples, as we have described before, the preparation, that is the meat, the pellet, and the tenfold, and we do metabarcoding, you can see that the actual ecology is quite stable. So nothing is changing from the ecological point of view, and this is something that is expected, right? Because we had a sample that was stored at -80, so by looking at the DNA, I would not expect that there was a big difference in terms of the presence of the species. Here you have another elaboration of the data that we obtained, that are expressed as delta log in the case of the viable count. So here you see how the populations are actually moving, increasing or decreasing. And again, we had a quite nice stability, especially for the sausage A. And this one that you see here on the bottom of your screen are the differences in terms of relative frequency for the main species that we found by the metabarcoding. And here you start to see again, it's actually, you don't start to see, but it is reconfirmed that there is a different trend between the sausage A and the sausage B, because in the sausage A, there is a quite nice, stable situation in terms of ecological setup. For the sausage B, we started to have more deviations or more changes because we had some populations that were decreasing, some other populations that were not as stable as in the sausage A. And here is the volatile profile. So in the case of the volatile profiles, you can see that there is actually a quite stable situation. So all the main volatiles that we were able to find in the sausage at T0 are actually confirmed to be present also at T12. But again, this is expected because we didn't do anything else than just store a sample at the -80. Okay, so metabolically here, we don't expect to see any differences because everything was blocked at -80. The next one is a little bit more challenging because it was the propagation. So what we have done was to use these three different media, as I was telling you before, PHI, meat broth, and nutrient broth. This is for the sausage A, and this is for the sausage B. Those are results in counts after the propagation of 24 hours, okay? And so you can see that basically the different populations were behaving differently in terms of their growth based on the type of liquid medium that we were using. I'm not going to be describing in detail the trends here because, again, I'm going out of time, but I just want to say again that probably in all this exercise, the population that suffered the most was actually represented by the yeasts. And you can see that in all the propagation media that we used, both in sausage A and sausage B, the yeasts were the one that actually could not grow or could not reach a level that could be somehow used for the further inoculation, okay? All those samples were subjected to 16S rRNA again. And so here you can see how the different populations moved ahead during the propagation. Here you have samples stored at 0, at 3 months, 6 months, 9 months, and 12 months. And for some of them, we were able to reach a kind of stability in terms of the populations, but for other ones, we had problems related to this Enterococcus that, as you can see, not present in the original sample, but was actually present a lot of times. And we don't know if this was matter of contamination or this was matter of something related to the sample that could actually enhance the growth of these microorganisms. And here is again relative abundance differences. So the situation is quite stable, and independently from the storage time, we were able to reach a certain number, because here again, we are talking about numbers here of the populations and relative abundances, so we were happy with that. Something that was not confirmed for sausage B. Sausage B, from the very beginning, we have seen also before, it was behaving quite strangely. And if you take a look at these maps, taxonomical maps, really, it is very, very confusing. And also in terms of relative abundance, we have a lot of populations that are growing and they are not actually making any sense. For this reason, what we have done was to discard sausage B from the further steps of the experiment, and we focused on sausage A. And what we have done was to really go into the production of fermented sausages. So we have used these propagated microbiomes. As you have seen before, we inoculated them in fresh meat with fat, with salt, with pepper, and we fermented the sausages for 15 days. Here you see the counts again. So are quite let's say representative of what is happening during the fermentation of the meat. So lactic acid bacteria and also the coagulase negative cocci that are growing, the enterobacteria that are decreasing. So the situation is quite stable and actually quite expected. And if we go into the taxonomical maps, you can see here that, again, independently of the microbiome that we have, the propagated microbiome that we had inoculated, because this is the one in BHI, this is the one in the meat extract, and a different is the meat here in the BHI and in the meat extract. At the end, at T15, the ecologies are quite stable. Are different from the backsloping and from the control. And here I like to highlight how the control ended up with a microbial setup that was quite different from the previous one, with some microorganisms that were actually were not characteristic, let's say, or were not classically found also in the normal fermentation, in the spontaneous fermentation of fermented sausages. Okay, and here is the slide that I showed you before about the volatilome. I can tell you that I was the one that tasted those fermented sausages at the end of the maturing, and you could really see a difference, or you can taste a difference between the control, the backsloping, and the other one. So this is something that we were happy, because the inoculation of the propagated microbiome could be done and could become a feasible solution in terms of reuse of the microbiomes. And just one last shot about, again, MAGs, because this is something important, in my opinion, to demonstrate that the storage of the microbiomes could be also preserving the biodiversity in terms of strains that we had in our sausages. Here you see like an overall picture with the different MAGs that we were able to reconstruct from the metagenomic libraries. But even more importantly, you can see here that, especially if we look at the MAGs that we were able to reconstruct about Lactobacillus curvatus and sake, you can see that those that were able, those that were present at T0, let's focus for a second at this one. So this was a MAG reconstructed at T0 that actually was very close to other MAGs that we were able to recover, not only from the propagated, but also from the sample that was stored at the very beginning. So again, the system or the protocols somehow are working, and they are able to preserve the biodiversity. I'm closing very fast because I've been already talking for a long time, but you can see here some of the conclusions. So it is possible to store microbiomes when it comes to fermented sausages. And my colleagues, I see that there are several ones that are connected here that contributed to this VP, can say the same also for the foods or the systems that they have been investigated in the project. One limiting factor is about complexity of the ecosystem. So if the ecosystem is very complex, then we have a problem, because that is going to be very complicated to be maintained and to be reproduced during the propagation. This is something that we need to recognize. And as a future perspective and challenges, this is something that I believe that this platform and this group of people that is following this seminar is actually the very correct one to be starting thinking about addressing this challenge, because we have become very good in storage and preservation of single strains. And so we have now a lot of guidance, protocols, indications on also which are the data that we need to collect for the storage. For the microbiome, we don't have anything. So we need really to put efforts in order to define what is the minimal data set that is necessary for the microbiome storage. I want to believe that in the future, our collections will not only have strains, but we'll also have microbiomes stored. But in order to do that, again, we need also validate protocols for storage, for propagation. We need to engage with industries and give them really or show them really the opportunity of using microbiomes. And then we need to be very flexible in the approach that has to be used when we need to work on single cultures, or when we need to work on microbiomes. And here I just want to close with one slide, because to me it's very important also the messages that we are delivering as a group, because probably we have been focusing too much on the use of synthetic communities, because this was the first term that was used also in science to describe the complexity of the ecosystems that somehow we are addressing. But probably this synthetic need to be reconsidered, and that's why I was part of an effort to really think about trying to switch from synthetic communities to defined microbial communities, in order to give a clearer message also to the stakeholders, to the industry, to the consumers, because again, we don't want to get into the trouble that we had in the past with genetically modified organisms, for instance, because that was a very, very important pitfall, in my opinion, that we had to face. That's it. Thank you very much. I mean, I know that I've been, I went a little bit over time, but I hope I was not boring, and I was able to keep your attention up. Thank you very much.